do you see that in edit mode?
Hi everyone, I'm going to share my screen now. Sorry for the delay. So thank you, Mahalaya, for organizing this webinar series for us. I would like to welcome everyone, including the speakers, the panelists, and the audience who are here today to listen to our day 2 of the Nucleic Acid Working Group-led series of webinars. And today's discussion is around the lipid nanoparticles and messenger RNA therapies. I would like to quickly go over some of the key concepts that we have covered in this nucleic acid working group. And if you joined us yesterday, we covered this siRNA and antisense oligo-based modalities, and we looked at both the translational ADME, DMPK biodistribution, as well as bioanalytical and immunogenicity components, and provided good best practice recommendations, as well as the gaps. Today, we are continuing on with Dr. TALG from Vertex, who led this second series, and he's going to talk about, and the other speaker in the panel will talk about the lipid nanoparticles and RNA. I'm going to hand over the slides now to Tal, who's going to introduce the scope of this webinar series.
Hello, I'm just speaking using Viva's laptop.
Looks good.
Okay, cool. So this particular presentation and webinar will focus on the application of lipid nanoparticle encapsulated mRNA as protein replacement therapy for rare genetic diseases. Other diseases such as cancer is not within this the scope of this presentation. And lipid nanoparticle encapsulated mRNA, as you all know, is a novel modality, and the entire industry has very limited clinical experience with this particular modality. It's also evolving very quickly, and so therefore this presentation is based on the current understanding of all team members and the corresponding IQ member companies. So here is the table of all the team members. Myself is the lead, Jesse, co-lead from Merck, Pedro, Manuela, Joanna from Bayer, Wen from J&J, Ming, Yun, Aqua, Jason from Moderna, Max from Novartis, Oleg from Vertex, and Lina from Vertex as well. Sorry, Oleg from Regeneron. And so today we are going to have 5 speakers. Viva has a lot of experience with bioanalytics. She's currently the executive director at BMS, and myself with a lot of experience on genetic therapies, including mRNA therapy. Marcus Wallace, currently the global head of biotransformation at Novartis. Pedro, currently expert for new modalities from DMPK at Bayer. And lastly, Ming Liang, from clinical pharmacology and pharmacometrics at Moderna. So I'm now going to turn over to our first speaker, Marcus.
Marcus, we can't hear you.
My name is Marcus Wallace. Can you hear me now?
Yes, thank you.
Okay, cool. I'm global head of biotransformation at Novartis. Over my years, I've been in touch with some of these modalities like antisense oligonucleotides, siRNAs, as well as some RNA vaccines. So today I will present the view of the team on the DMPK needs for the development of these new modalities. So I just share my screen here. Okay. So in the next couple of minutes, I will just go quickly over the regulatory landscape, And then we'll also cite some literature. The views in this presentation are not all from us. A lot of them have been published, especially in regards to biotransformation. I really propose you to read this paper from Verweg et al., which has been published last year. So the reference is listed here. I will go quickly over some regulatory guidance for the characterization of excipients because it's very important. And then we quickly go over the performed ADME studies for the registered RNA vaccines, Comirnaty and Spikevax. And at the end, I will close with some general recommendations. So how does a regulatory landscape look like? So yesterday, a little bit more clear defined. Some are developed as small molecules, some are developed as biologics. But for example, inclisiran from Novartis has been developed as small molecules, so an NDA has been filed. So for these, you have kind of a mix of regulatory guidelines from small molecules as well as large molecules. Because of this, some of these IND packages of these modalities, they look very close to the ones of small molecules, depending if you file in BLA or an NDA. For RNA vaccines, this is kind of a complete new situation. Although by definition, the concept of the RNA vaccines to generate a protein falls in the area more of gene therapy. I think the current registered vaccines, they have been registered similar to infectious disease vaccines. So the FDA has, for the COVID, released their own COVID-specific guideline. And in Europe, the EMA always refers to the general WHO guidelines for infectious diseases and vaccine generation. So these two vaccines are covered under these guidelines. Looking forward, we all know that also RNA vaccines are developed for the area of oncology. I think we don't have a precedence yet, so this has been really regulatory handled. So we're looking forward to this. As I mentioned before, it would fall by definition under gene therapy, but let's see what we can expect in the future. Let's go to the next slide. So one of the key studies which need to be conducted for these antisense oligonucleotides and siRNAs you have discussed yesterday are the biodistribution studies, because you want to know in which tissues your entities more or less end up and where they distribute to. On the top part here, you have shown just a schematic of an siRNA. So you see you have a double-stranded construct of 21 bases. So that's the example of patisiran. And you see you have chemical modifications in these strands. And that's, I think, also one reason why we need to kind of follow these chemical modifications to see where they end up. So for the classical RNA therapeutic like siRNAs and oligos, biotransformation studies always have to be informed, at least according to the EMA. For the FDA, they're a little bit more moderate, so they clearly say it's just needed if you do changes to the delivery of your entity, meaning the backbones, the vector backbones, the formulations, the route of administrations, and if you change the dosing schedules. So the design of your dependent, so you can either kind of track your molecule yourself, the molecule itself, by labeling it, for example. We will discuss this later, or you can just also track the delivery system, because I think for siRNAs and ASOs, the general perception is they are not taken up actively by the tissue unless you have these GalNAc modifications, for example. But if you just have a natural siRNA, the permeability should be low, so the formulation should actually drive the distribution of these entities to the tissues. In certain cases, these biodistribution studies can be avoided. We will talk about this in a few minutes. But I think what is important to highlight is, even for these entities, if you develop your therapy with new formulation components, these new components need to be characterized well in clinical studies before you go to the clinic. For the vaccines, you do not really require these PK and distribution studies. So this is shown here at the right-hand side. So I didn't draw a sequence, but you have about 4,000 bases, single-stranded, and you do not have a whole bunch of chemical modifications. So usually you have just these uracil replacement by the N-methylpseudouridine, and this is kind of a natural modification. It's not really considered a chemical one. But here also the exceptions play along. So if you use novel adjuvants, formulations, or entities in your formulation, you also would need to characterize them and show where they distribute. For the registered products from Pfizer and Moderna, so no biodistribution studies were performed for the final LNP formulation. They used surrogates before to characterize their molecules, which we will discuss in a second. Also, the FDA guideline states that this COVID vaccine platform technology can rely on existing tox data of other platform molecules, which is really great. And so far, we saw, I think one is approved, one has, I'm not sure if there is still a conditional approval or if it's fully approved in Europe. But so far as we know, no additional in vivo data had to be submitted. So that means that this approach has been more or less accepted by these two health authorities. So this slide just shows you the performed biodistribution studies for the two RNA vaccines. So for the Spikevax, we have a Cuba study performed. So we will discuss a little bit in a minute what Cuba is. And in addition, parts of the sequence were tracked by a branch DNA assay, and Pedro will discuss later about this in his presentation. For Comirnaty, also the molecule has been tracked by Cuba, or the distribution studies have been performed with Cuba. So here we also have a distribution with a radio label, and I think it was in this case also on the one of the formulation components, not on the RNA itself. And in addition, other formulation components were tracked by LCMS as well as the sequence. So Cuba is a known technology for, I would say, most scientists who come actually from the small molecule world. So these studies, quantitative whole body autoradiography studies, they are performed standard for small molecules in later development. And one reason is you want to see the distribution of your compounds in the tissues to also estimate the dosimetry burden when you dose later radio-labeled material to humans. For preclinical studies, nevertheless, it's also very helpful to understand how your components are distributing. Usually Cuba studies need to have a radio label, so they have, you need to label the molecule of interest with a radio label. For small molecules, usually 14C is the preferred choice. For larger molecules, tritium is often the isotope of choice because it has a higher specific activity, but nevertheless, it also has its disadvantages. For example, tritium is easier to cleave off from your molecules. So you might have release of tritiated water, which then interferes with your distribution. It might be not that visible on these slides because these slides are usually generated once these tissues are completely dried and taken out and prepared. But nevertheless, if you lose your label, you cannot really track where your molecule of interest is going. So for the RNA vaccines, you have different molecules to consider. You can label either your RNA sequence. You can label some of the delivery vehicle, like the LNP particles. And I think in this case, which is shown here, I think the cholesterol of the LNP has been formulated. Why, you might say, is that a valid approach? As I mentioned before, just natural RNA will not have any passive permeability, so the distribution will mainly be driven by the isotope. Not by the isotope, will be driven by the formulation. So if you label the formulation, you get a good picture of the isotope. Coming back to the formulation, there's really specific FDA guidelines for the evaluation of these pharmaceutical excipients. And if you kind of introduce a new ionic lipid in your formulation, you need to consider that you also need to, in most cases, perform a defined set of ADME studies, meaning absorption, distribution, metabolism studies, to have a good understanding of your molecule before you enter human clinical trials. So I've extracted this from the guidance, and the guidance are cited here. So if you want to read this up, strongly recommended to read it. So which studies have been conducted actually for Comirnaty? So for Comirnaty, they have actually a new excipient in their formulation. It's the aminolipid and the PEG lipid. And I think these lipids have been characterized. So there have been pharmacokinetic and elimination studies performed in rats, but for these lipids, not radiolabeled, they have been tracked usually with LCMS. But I think plasma, liver, urine, and feces were analyzed for the fate of these lipids in these studies. Metabolism was also investigated for these new formulation components, not for the RNA itself. So they were examined also in vitro studies using blood, liver, S9 or hepatocytes from different species as well as human. So the excipients also have been tracked by LCMS without any radiolabel. In addition, the in vivo metabolism was also examined in rat plasma, urine, and feces, as well as liver from rat pharmacokinetic studies, but not with the RNA, which has been applied in the end. So, but they used a surrogate molecule, luciferase encoding RNA, which was formulated with the same formulation to derive this information. The distribution studies, as I mentioned before, was done by Cuba, and for these formulation studies, actually the cholesterol in the LNP formulation was radiolabeled with tritium. This is a valid approach because I think the formulation is driving distribution into the tissues. This has been not done with the final formulation, but also with a surrogate molecule for the RNA sequence. As I said before, no traditional web pharmacokinetic and biodistribution studies have been performed with the final candidate at the end. And then for the excretion of the two novel lipids in the formulation, these were studies simply by PK studies. No radiolabeled studies were performed to determine the fate. For Spikevax, it looks similar. So no dedicated ADME studies were conducted for the RNA sequence per se. There have been biodistribution studies performed with the surrogate molecule. It's called the mRNA 1647 in a non-GLP tox study. So the tissue distribution was determined. The distribution of the lipid nanoparticle content was determined. And I think what was very relevant here is that they did not perform all the studies with these lipids they used in the formulation. There have been similar lipids already registered before for certain other products, and some of the studies could be referred to these lipids because they were structurally very similar, and I will discuss this also on the next slide. But nevertheless, I just want to highlight also here no dedicated studies for the RNA sequence have been performed for the final RNA product, which have been gone into human. So now let's come back quickly to the distribution of these LNP components. So the distribution of a novel lipid component has not been extensively studied in these studies. But as I mentioned before, studies have been previously conducted with a close analog, which is structurally very similar. So at least in the final registration dossier, the company could actually refer to this data and to bridge the results. So this is also another way to qualify your new components. But I would say, I will also mention at the end, it's always recommended that you discuss the strategy with the health authorities before you execute. The quantitative whole body autoradiography, which was performed for this lipid 5, confirmed that there was no prolonged retention of this in the tissues. And because of the structural similarity between this lipid 5 and SM102, it was assumed that these lipids distribute similarly and are similarly metabolized. So what would be our final recommendations based on these observations we have made in this previous successfully registered products? So first of all, for the mRNA part, radiolabeling is not necessary. I mentioned before, the RNA itself is usually not chemically modified. It also has limited passive permeability. So cleavages would not, released RNA will be quickly deteriorated in circulation. So I think there's no need to follow up on this. But similarly, no biotransformation studies are necessary for the mRNA part. But in the future, if you start to also modify the mRNA with chemical modifications, this of course might shift the picture. For the LNP particles, it would be important that new formulation components are thoroughly characterized. So if you introduce new lipids in your formulation, you need to conduct the relevant ADME studies. But nevertheless, as I showed before, you can also refer to approved lipids. So if you use lipids in your formulation which have been improved before, this can be used to mitigate some ADME studies or even waive some, and also to avoid radiolabeled studies. In general, we feel that biodistribution with LCMS could be sufficient. Radiolabeling, of course, can add some value, but it's not a must. Nevertheless, we strongly would recommend that you discuss your plans, how you want to characterize your formulation and your RNA with the health authorities, because I think there's limited examples there. So if you have a completely new indication, or you have some other chemical modification, for example, in your RNA sequence, then this picture might change. So thank you very much for your attention. And now I would like to hand over to my colleague, Pedro.
I'm Pedro Moraes. I work at Bayer Pharmaceuticals. I work in Wuppertal, Germany. I'm an expert for new modalities, including mRNA LNPs. I've been working in oligonucleotides and RNA for more than a decade. And I am now at DMPK at Bayer at the Bioanalytical department. So in this section of today's IQ webinar, I will discuss bioanalysis of LNP encapsulated mRNA therapies. In this presentation, we will start with some general considerations about mRNA. We will then briefly outline the different bioanalytical validation requirements at the different development stages. Subsequently, we will take a look at different bioanalytical platforms for mRNA and examine some of the challenges regarding RNA extraction and RT-qPCR, which could have an impact on biodistribution studies. We will then briefly review some primary design considerations and present the ddPCR as an emerging absolute quantification method for mRNA. Finally, we will summarize the main lessons from the current bioanalytical lessons for mRNA, describe the current regulatory guidelines in the landscape and documents that could provide some additional guidance, and we'll briefly describe some potential immunogenicity assessment strategies for LNP encapsulated mRNA. But first, let's start with the elephant in the room. So RNA is a large molecule that is susceptible to degradation by ubiquitous nucleases and therefore has its intrinsic bioanalytical challenges. Also, it is important to realize that a significant portion of the current mRNA LNP programs under preclinical and clinical development have uridines in the mRNA replaced with pseudouridines. So this is an RNA modification that improves mRNA stability against nucleases, but also modulates the intrinsic immunogenic character of the mRNA. Fortunately, this RNA modification does not have a significant impact on the bioanalytical strategy because pseudouridines or N1-methyl pseudouridines, which are also used, are read as uridines by polymerases typically used in the PCR. The current recommended approaches of bioanalysis of mRNA are the branched DNA assay and the RT-qPCR reaction. As outlined by my co-presenter in this webinar, Marcus, the known bioanalytical platforms in currently approved COVID-19 mRNA vaccines are QWBA and IVIS for Comirnaty and QWBA and branched DNA assay for Spikevax. So at different stages of mRNA drug development, there might be different bioanalytical validation fit-for-purpose methods. As recently discussed in an industry-wide white paper published in 2022 and referenced here, for mRNA, informative PK endpoints are recommended for the early unregulated preclinical stages, during which assays can be qualified. In the subsequent regulated preclinical phase, biodistribution data generated in a single species is recommended for IND submission. Blood analysis can be performed with a validated or a fit-for-purpose method, and tissue would require a qualified method. Finally, for clinical phases, the systemic exposure of mRNA may be the primary endpoint when the expressed transgene protein is not. Further, the bioanalytical assay can be qualified or validated based on the intended use. So there are several methods that enable the bioanalysis of mRNA, and we will briefly examine here. Our main focus in this webinar section is, of course, the RT-qPCR, but we will provide an overview of techniques such as bioluminescence imaging, or BLI, the branched DNA assay, and the digital droplet PCR. So one of the emerging techniques that can enable the visualization of the transgene encoded by an LNP encapsulated mRNA is bioluminescence imaging. Well, typically the mRNA sequence should encode a bioluminescent gene, such as a luciferase, which is administered to the animal, and in the presence of the substrate for this luciferase, like luciferin, a bioluminescent signal can be generated and observed in different parts of the animal. So despite the fact that this flexible and non-invasive technique enables the direct interrogation of the translation kinetics and the delivery of the LNPs, which is important, it is not as sensitive as the QWBA method. Obviously, when a bioluminescent gene is used instead of the therapeutic one, as an assumption has to be made that the sequence doesn't have an impact on biodistribution of the LNP encapsulated mRNA. Another important assay that is gaining traction for bioanalysis of mRNA is the branch DNA assay. One of the biggest advantages of this method is that it does not require RNA extraction from the tissue, meaning that the direct measurement can be made in lysates without the need for PCR target amplification. Instead, it is the signal that is amplified, and this can be achieved with a series of probes that hybridize to the target RNA and then become the template for additional hybridization with pre-amplifier probes. So ultimately, a fluorescent or a chemiluminescent reporter molecules can bind to these complexes in a manner that is proportional to the amount of the target RNA of interest, enabling quantification. But the main focus of this webinar section is, of course, the RT-qPCR. So this is the reverse transcriptase quantitative polymerase chain reaction and enables the detection and quantification of the mRNA. So in this reaction, the mRNA transcripts can be detected in a very sensitive manner, taking advantage of the sensitivity of the PCR reaction. In the RT-qPCR, the RNA is copied first into a cDNA by reverse transcription via a reverse transcriptase, which can synthesize cDNA with sequence-specific primers or random primers. And then the cDNA of interest is amplified with the conventional PCR reaction and a DNA polymerase. Both reactions of the RT-qPCR, meaning that the cDNA synthesis and the PCR amplification, can either be done in a single step or in a two-step assay. So before the the mRNA is detected and quantified by RT-qPCR, it needs to be extracted from biological samples. So due to the ubiquitous nature of nucleases that degrade the RNA, this process requires some considerations. Well, here's some examples. A commercial solution that protects the RNA from degradation can be added to tissue following its homogenization in order to prevent degradation. It's also important to note that the efficiency of the extraction can sometimes vary in different tissues or matrices. And also, different extraction methods can be used, from the standard phenol chloroform extraction to silica or bead-based methods. We will discuss it in a subsequent slide. The sample characteristics can also impact RNA extraction efficiency. For example, the extraction of RNA from tissues with a high lipid content may require additional steps, such as the removal of the lipid fraction. But anyway, what is important in the end is to control the quality of the extracted RNA, which can be measured via the RNA integrity number, calculated with a bioanalyzer or similar equipment. But also UV analysis is very useful of the extracted RNA in a nano drop device, for example, that can help us assess how much protein in or salts can be present, which could potentially inhibit the PCR reaction. So as mentioned, there are different RNA extraction methods which can impact efficiency and thus quantification of the mRNA of interest. Each can have advantages and some inconvenience. For example, the classical method based on phenol chloroform extraction can be highly efficient, but has a low throughput and is time-consuming. On the other hand, beads-based methods or the use of silicon membranes can be automated, which are suitable for a bioanalytical lab, but can have a lower efficiency. So considering the challenges of RNA extraction in some tissues, the use of spiked-in controls can enable determination of extraction efficiencies. Now we will discuss some considerations regarding the RT-qPCR in the bioanalysis of mRNA. As for the assay setup itself, as mentioned in the previous slide, the RT-qPCR can be executed in a one-step or a two-step manner. A one-step setup reduces contamination and pipetting errors and is a more streamlined process, especially for large experiments. On the other hand, the two-step setup could provide more flexibility, especially for the optimization of the RT-qPCR itself. As for critical reagents, it can be said that the selection of primer is crucial for the RT-qPCR. For the generation of cDNA, they can be random or sequence-specific, and in regards to the detection of the amplified PCR products, this can happen either via intercalating dyes or sequence-specific probes such as TACMAT. As for the calibrators, they can be either DNA or RNA-based. The DNA ones are obviously more stable and reliable, but they cannot account for the reverse transcriptase efficiency step. This can be solved with an RNA reference molecule, but then RNA is less stable than DNA and more prone to degradation if not handled properly. The proper selection of housekeeping genes is also, of course, very important in the relative quantification of the mRNA of interest. As for the analysis of the data generated in the RT-qPCR, it's important to keep in mind potential impact of matrix effect and or the presence of PCR inhibitors that can come from the extraction process itself. As for assay specificity, there is a recommendation from the FDA from 2006 for the use of spiked-in DNA controls. For relative quantification, a housekeeping gene is used instead of a spike-in control, but ideally, the expression of this housekeeping gene should not be affected by the treatment, and its amplification efficiency should mimic the target genes. For absolute quantification, typically the mRNA is reported as copies per microgram of total RNA and not normalized to tissue mass. And so what about the challenges in the use of the RT-qPCR for mRNA distribution? Well, typically different tissues need to be collected and the RNA needs to be extracted from them, which often can require different extraction techniques because different tissues can have more or less RNases or be more or less difficult to physically homogenize. Further, tissues can vary in size and the presence of potential PCR inhibitors, as well as inhibitors of the RT reaction as well, or even RNases. Therefore, it is not uncommon to see higher CV values in some tissue samples where these challenges are more acute. So, as mentioned before, primers are one of the most critical reagents for RT-qPCR. In order to have a target-specific amplification and proper quantification of the mRNA, there are general guidelines for primer design, which we will outline here. First, for the primer sequence design, there are software available tools that can generate target-specific forward and reverse primers. It is advisable to blast the obtained primer sequence for any potential cross-reactivities while considering the use of different species in preclinical and clinical studies. These software tools should select primers that avoid dimerization and self-folding. The PCR efficiency should be between 90 and 110%, although as low as 85% could be acceptable for early studies. Ideally, there should be a similar PCR efficiency between the target amplification and a housekeeping gene in the relative quantification efforts. As for sensitivity and specificity, it can be assessed through the melt curves to ensure that only one PCR product is generated. Finally, it would be recommended that whenever possible, select primers. The primers are selected that can be used across preclinical and clinical studies. So we have covered so far the RT-qPCR as an important tool for mRNA distribution studies. However, this technique still has some drawbacks, such as the presence of PCR inhibitors that come from the extraction of the RNA from the tissue, And also the fact that if we are looking into very small amounts of target mRNA, they can be masked if the total RNA is much higher. So this is where the digital droplet PCR, it's a very promising technique that will probably establish itself in the future as an improved absolute quantification method that is more sensitive towards the target and less sensitive towards PCR inhibitors. Briefly, the digital droplet PCR does not occur in the standard solution, but rather in thousands of droplets that are generated. Then a Poisson statistics can enable the absolute quantification of the target of interest. Therefore, this is a method that has improved sensitivity, precision, and reproducibility, but it's not it's a little bit beyond the scope of this presentation, so I will not dive further. Okay, so in this slide, we have a very nice summary of the different bioanalytical techniques covered for biodistribution of mRNA. So starting from the right side, the IVIS method, it's obviously an interesting option that is non-invasive and also enables longitudinal studies in the same animal, which is really nice, and enables also, of course, a direct visualization of the surrogate LNP encapsulated mRNA transgene, but it's limited to reporter genes and requires administration of the substrate of the luciferase mRNA to the animal. Next, we also covered the branch DNA assay, which is another interesting method, and it's crucial to say that it does not require RNA extraction. So for tissues where RNA extraction is tremendously difficult, it might be a very interesting option. But despite the advantages, it's a method that is reliant on vendors and target-specific reagents. Then we have the PCR methods, especially the RT-qPCR, that offers flexibility and sensitivity. The RT-qPCR is the gold standard with several guidelines already published and available. However, for absolute quantification, it requires a calibration curve and is sensitive to the presence of PCR inhibitors. These drawbacks can be potentially addressed with a digital droplet PCR, which can be truly quantitative. However, this technique is not yet fully validated in a regulated environment and requires relatively expensive equipment and reagents not available in all bioanalytical labs. Now we're getting towards the end of this section of the webinar. So in this slide, we provide a list of the current regulatory guidelines for mRNA distribution studies. So the overall recommendation is for use of the RT-qPCR, although the criteria are not yet fully defined. So in COVID-19 mRNA vaccines, the branch DNA assay and the BLI was used in biodistribution studies, obviously something important to consider, but they have their limitations, as we briefly discussed. For mRNA detection in RNA therapeutics, there are EMA, ICH, and IPRP guidelines available for RT-qPCR, and the ICH S12 document also acknowledges the digital droplet PCR as an emerging technique, although it is not yet fully validated. So before we conclude this section of the webinar for bioanalysis of mRNA therapeutics, I would like to briefly provide some potential immunogenicity assessment strategies for LNP encapsulated mRNAs. For the immune response, the ELISA, or enzyme-linked immunosorbent assay, is the recommendation, especially for detection of antibodies that could be directed towards the expressed protein, but also, as we know, for assessment of anti-PEG antibodies. So one of the ELISA formats displayed here on the left side of the slide is the bridging ELISA assay, which is a type of sandwich ELISA, which enables the detection of antibodies binding to a first immobilized transgene protein to the ELISA plate surface. And then a labeled version of the transgene can be used to bind to the anti-transgene protein antibodies and enable their quantification. Regarding immunogenicity assessment of the cellular immune response on the right side of the slide, the ELISpot or enzyme-linked immunospot assay is recommended. In this assay, the cellular cytokines potentially produced by antigen-stimulated T cells in response to the transgene-expressed protein can be quantified with a surface-bound capture and then detection antibodies. So, to conclude this section of today's webinar, here are some general considerations that you should keep in mind for bioanalysis of mRNA. So, we need to be aware of the limited experience so far. So, most of the guidelines and information available coming from COVID-19 mRNA vaccines. Therefore, we believe that a case-by-case analysis should be performed for therapeutics, for bioanalysis of novel mRNA therapies. Then we have also seen the different pros and cons of the different bioanalytical methods for mRNA. And we think that the use of orthogonal methods should be encouraged. For example, analyzing the expression of the protein as well as the mRNA. We also seen that the immunogenicity assessment strategy can be very important in parallel with safety and efficacy studies. And finally, this field of mRNA therapeutics is clearly in its early stages, and therefore a solid bioanalytical strategy will need to consider multiple variables such as data analysis, disease indication, patient population. So with this, I conclude this section of the webinar. I would like to acknowledge Lena Lu from Vertex Pharmaceuticals, who helped me with the preparation of this slide deck, but also my colleague at Bayer, Manuela Braun, who also helped me. I thank you for your attention, and now let's move to the next section of this webinar.
Can you all see my screen?
Yes.
So I will cover the clinical and quantitative pharmacology considerations for lipid nanoparticle encapsulated mRNA therapy together with Ming Liang from Moderna. Okay. Sorry about that. Here is the outline. So I'm going to give a quick mRNA and LNP intro, followed by clinical pharmacology study typically required for this particular modality, and then early stage clinical study design considerations for this type of modality, and briefly immunogenicity considerations afterwards, and lastly, quantitative pharmacology applications, and together with a case example, mRNA 3927 in propionic acidemia, that's going to be covered by Ming from Moderna. So as you all see, this is a typical structure of mRNA modified and self-amplifying, and typically there's a 5' cap, 5' untranslated region, open reading frame, 3' untranslated region, and a poly A tail. And mostly thanks to Kathleen Carrico and Drew Weissman from UPenn on their discovery of using pseudouridine to replace uridine. Therefore, we can have reduced immunogenicity for mRNA. And for lipid nanoparticle, as you see, here is a typical structure of lipid nanoparticle. And typically, you have negatively charged mRNA in the center, circled by the ionizable cationic lipid. And molar ratio-wise, typically ionizable cationic lipid is about 50% of the entire LNP, and then followed by cholesterol, about 40% also. And then helper lipid or DSPC, about 10%. And lastly, PEG lipid, about 2% also, typically at the outer shell. And this is just a hypothetical, composition of the mRNA and LNP. And as you see, this LNP is typically widely used for delivery of nucleic acids. It can be used for siRNA, such as patisiran, mRNA, and this structure has about 100,000 molecules and about 100 nanometer in diameter. And the uptake typically is mostly by liver, followed by spleen and other organs. And for immunogenicity, as you see, typically the PEG lipid as well as target proteins can give you immunogenicity issues. And here is a structure of the lipids for the two COVID vaccines. On the top, we have the one from Pfizer and BioNTech. At the bottom, we have the one from Moderna. And as you see, for ionizable cationic lipid, typically there's a tertiary amine out there and a couple ester bonds. The PK typically is between 607, which is responsible for the disintegration of the entire LNP and therefore release of mRNA in the endosome. And two helper lipids, they are endogenous, DSPC and the cholesterol. And lastly, the PEG lipid to the right. And one thing I would like to call is that the PEG lipid here, as you can see, the molecular weight typically is about 2,500-ish. And this is much smaller than the typical PEG in the PEGylated protein, for example. So therefore, it's not a good idea to generalize on a number of things, such as immunogenicity to the PEG from the PEGylated protein for mRNA. Okay, now let me move on to talk about the mechanism of action. As you see, when we have the LNP encapsulated mRNA, and after administration, either IV or local delivery, such as pulmonary delivery, it gets into our human system, and the LNP can protect the mRNA from degradation by all kinds of nucleus. And tissue-specific mRNA delivery typically mediated by LNP and mostly goes to liver, like I said. And a protein called apolipoprotein E in the plasma can be bound to the outer shell of the LNP, the PEG lipid, and then that is responsible for the uptake through LDL receptor. Although I would like to call that there are some publications stating this is not the right mechanism for uptake. But I would say this field is quickly evolving, so we will see. And like I said, endosome release is a critical part for mRNA delivery. Once mRNA gets to the cytosol, it will go to the ribosome and then give you the target protein, either a secreted protein or intracellular protein or cell membrane bound protein. So again, their location will determine the immunogenicity implications, and obviously you need different assays, when the protein are localized either membrane bound or intracellularly or secreted. So then, as you see to the left, these are the typical clinical pharmacology studies required for small molecules. But in general, most of these things do not apply. But for this type of modality, you need to monitor the immunogenicity, like I said, and also the PK for novel lipid, particularly the accumulation of novel lipids. And so then I'll move on to talk about the study design considerations for early stage clinical trials for this type of modality. I don't want to talk about late stage because there's none, particularly for rare genetic diseases. And first of all, the sponsor can use single arm studies, but it should generally focus on mathematical needs. The study population, we can initiate the first in human studies directly in patients based on mathematical needs without healthy volunteer cohort. And placebo subject may not be needed based on mathematical needs and or prior experience with placebo effects in the particular disease. And primary endpoints, typically safety and tolerability, and sometimes PK/PD. Secondary endpoints PK/PD if it is not included as one of the primary endpoints, and biomarkers, obviously, and or early efficacy endpoints. In terms of the dose, which is a particular important thing for clinical pharmacologists, the top dose may be based on tox data, such as human equivalent dose HED of monkey NOAL. And the reasonable safety margin can be applicable, such as 1.2, 25-fold, 1.5-fold, or 2-fold. The starting dose may be based on the safety approach, such as the tox data, like I said, human equivalent dose of monkey NOAL divided by a safety factor can be 10, or prior human experience with similar construct. And the starting dose may also be a minimum efficacious dose, or MED, based on the pharmacology data. And you can use a bunch of things as shown here, or modeling approach, such as translational PK/PD modeling. And typically, if you have orthogonal approaches that cross-validate the starting dose, that will be ideal. And sometimes you may use MABEL approach for starting dose. That typically is used for cancer vaccines, for example, or something that's supposed to modulate the immune system, particularly as an organist. But for rare genetic disease with unmet medical needs, you can use a minimal efficacious dose or MED approach. The dosing frequency typically is based on the preclinical data. And dose escalation scheme, for mRNA therapy for rare genetic disease, typically a 3+3 design or certain variants is being used. And anticipated tox certainly is a consideration for dose escalation. And in terms of the fold between different cohorts, and typically it has to be lower than or equal to 3-fold. So basically log escalation is generally considered too aggressive. And intra-patient dose escalation typically is not recommended. And the sponsor typically need to apply certain type of staggering between different trial participants or dose cohorts. And also the sponsor may apply sentinel dosing. And for dose escalation, like I said, 3+ design typically used in oncology. And if no dose limiting toxicity or DLT in 1 out of 6, that typically means you can dose escalate. If you see more than 2 or equal than 2 out of 6 DLT, then you have to dose de-escalate. Of course, this is based on a general principle, and for each individual program and case, it has to be considered case by case. And for dose limiting toxicity DLT, it is certainly to protect trial participants, identify the recommended phase 2 and phase 3 doses. And typically you may expect some immunogenicity, like I mentioned earlier, anti-PAC antibodies, anti-express protein antibodies, or interferon gamma secreting T cell response to express protein if it is an intracellular protein, for example, that may be expected. And also the sponsor may need to monitor the off-target toxicity, such as on the cardiac channel. And QTC study may not be needed, but based on the mechanism of action and prior experiences. Like I said, the mRNA is pretty large, and the molecular weight can be more than 1 million Dalton. So it has a pretty low likelihood to work on the cardiac channel. But if you are using a novel lipid within the LNP, then there is a chance that the novel lipid as a small molecule may work on the cardiac channel. So therefore, QT and QTC studies may be needed. And safety monitoring. So the duration of the safety monitoring for adverse events typically should be sufficient to cover the expected duration of effect. For example, 5 half-life for the novel lipid that potentially drives the AE. And 5 half-life for expressed protein that potentially drives the AE, for example. And also it depends on the information from preclinical studies, experience with similar products. And lastly, long-term follow-up may be required under certain circumstances, such as if you are dealing with a novel non-biodegradable lipid within an LNP, then you may need a pretty long-term follow-up. So I'll move on to briefly talk about the immunogenicity.
like I said, this entire field is evolving, and we are learning what kind of factors can contribute to immunogenicity. And as you see, product-related risks such as RNA tap modifications, lipid nanoparticles, or the expressed protein can all give you immunogenicity risk. And process-related risk can also give you immunogenicity issues, such as RNA fragments or empty LNPs. And third, treatment-related risks, such as dosing regimen, and route of administration. And lastly, patient-related risk, such as disease background, and the outcome of the risk determining the immunogenicity sampling and testing strategy. And also, I would like to acknowledge Manuela. She's leading immunogenicity team for this particular topic. And in regards to the immunogenicity testing strategy, as shown here, for preclinical studies, you can collect samples for immunogenicity assessment on a case-by-case scenario based on the outcome of the risk assessment. And also, you can analyze samples if required for interpretation of PK/PD and safety observations. You can assess antibodies against the PAG, the expressed protein, the cellular response against the expressed protein. And for clinical studies, you certainly have to analyze samples based on the outcome of risk assessment and non-clinical experience. So you can collect and hold if the molecule has a pretty low risk, or you have to collect and test if the molecule has a higher risk. And the sampling schedule, as shown here, typically you need a pre-dose sample. You typically need more frequent sampling at early stage clinical development, for example, first-in-human study, and less frequent sampling in later stage studies such as phase 3. And the sampling should appropriately cover the activation of inflammatory cytokines, complements, antibodies against PAG, target the expressed protein, and cellular immune response may be optional. And of course, you need to collect PK samples at the same time point for better interpretation and optimal timing depending on the RNA tap, LNP, as well as transgene protein. And for potential impact of the immunogenicity that's triggered by the LNP, you can have hypersensitivity reactions and accelerated blood clearance, for example. And for potential impact of expressed protein immunogenicity, this is particularly important in comparison to the enzyme replacement therapy. It really depends on the patient's background. All right, if the patient has been exposed to that particular protein or a protein with similar construct before, and also the impact on the safety and efficacy. So in general, I would say we only have very limited clinical experience with this topic, the immunogenicity, and with rarely observed LNP immunogenicity observations. And typically, if you do see LNP-related immunogenicity, it's not going to affect your safety, PK, PD, and efficacy. Again, like I said, this entire field is evolving quickly, and we all are learning. So then I'll move on to quickly talk about the quantitative pharmacology applications or modeling and simulation applications. Something that's specific to this type of modality is the time delay between the plasma PK and the PD outcomes. And typically target tissue concentration can, the predictions can be highly relevant. And in terms of the translation, you can apply allometric scaling to predict plasma PK, tissue PK, as well as PD in humans from preclinical species. And also it will allow, accommodation of different species. And that typically is used for first-in-human study dose projection. And in clinical studies, you certainly need to project the SAD, single ascending dose, as well as multiple ascending dose cohort. And also the dose can be based on the routes as well as frequency of administration. And you can also consider fixed dosing or body weight based dosing. And in terms of the modeling techniques, typically population PK/PD modeling, or KPD modeling if you do not have a PK concentration. and or semi-magnetic PK/PD modeling, which typically require strong bioanalytical support, particularly in concentration in the tissue. And here is just a rephrase of what I mentioned previously. And here, as you see, particularly for first-in-human dose projection, you can use the monkey NOAEL and apply a safety factor such as tenfold for your first-in-human starting dose, or translational PK/PD modeling. For clinical studies, you can start a dose at a minimum efficacious dose, particularly for rare genetic disease, and first-in-human study possible in patients directly without a healthy volunteer cohort, and also directly with a MAD cohort without a SAD cohort. And for novel lipid, you typically need to monitor the PK very closely. And for the dosing and dose regimen optimization, it can be done through PK/PD modeling. My colleague Ming will give you an example. And lastly, for these clinical pharmacology considerations, such as QTC, might be needed if any QT prolongation is observed. And also for human ADME study, typically not required for this type of modality. And for organ impairment, you may explore the impairment status as a covariate in your population PK model, as well as PK/PD model. For immunogenicity monitoring, like I said, you do need to monitor it, although you can take different strategy. And then I'll move on to, then I'll hand over to my colleague Ming.
Hello?
Yes.
Oh, can you hear me?
Yes.
Okay, so I'm sorry, I'm trying to set up. Hello, everyone. My name is Min Liang. I am a pharmacometrician at Moderna. So today I'm going to share an example of, hold on. I'm checking my, okay, so today I'm going to share a case study to show how we're applying PK/PD modeling work on the mRNA drug development. So the mRNA 3927 is encoded for an intracellular enzyme. And here we're covering both PCC and PCCB genotype. And this is an ultra-rare, life-threatening disease with unmet medical need. So for the patient, PCC enzyme lost function, which leads to accumulation of propionyl-CoA here, and along with the metabolites of CoA. We're clinically monitoring the metabolites of 3-hydroxypropionyl acid, 2-methyl citrate, ammonia, etc. And the accumulation of this harmful compound can lead to metabolic decomposition event. Reducing the metabolic decomposition event frequency is our clinical endpoint of mRNA 3927 treatment. So for the disease-related biomarker, that's the PD part we're going to talk about next. Here is an overview of our ongoing mRNA 3927 phase 1/2 study, currently enrolling patients 1 year and older. So far, doses up to 0.9 milligram per kilogram once every 2 weeks with a traditional 3+3 design. Our primary endpoint are safety and PK/PD. Our first in-human dose is an efficacious dose with very cautious dose escalation. The first 2 participants have to be at least 8 years old. And so far, there's no major safety concern up to 0.9 milligram per kilogram. For mRNA drugs, the PK model is modified based on the traditional compartmental PK model. As we all know, for classical IV infusion PK profile, the Cmax is normally observed around the end of infusion. So elimination is the dominant phase afterwards. For the lipid nanoparticle encapsulated mRNA drugs, we observed another peak, a local maximum hours, some even days post the end of infusion. So based on published siRNA on petro PK model, we modified the traditional compartmental model to catch a second peak. Here is shown on the scheme, the rapid tissue uptake of LNP, then the redistribution of LNP. So both compartment 1 and 2 are part of a plasma compartment. The total amount of mRNA is then associated with the protein synthesization. So the PCC enzyme activity is then restored to break down the accumulated propionyl-CoA. For the PD biomarker model, we are using classical indirect response model. Here's the one when I mentioned that there's a modified compartmental model. Here's the goodness of fit of mRNA PK model here. As you can see, the population predicted PCCA mRNA is not well described at the second peak, whereas the local maximum we observed after end of infusion. So please be note that the ongoing clinical study have patient ages range from 1 to 26 years old, and our infusion time is also varying based on different dose level between 1 and 2, actually right now 1 and 4 hours to minimize infusion-related reaction. So with the modified PK model, the individual prediction are well captured. So then including the LNP redistribution compartment, so the individual prediction is pretty well. Here's the challenge. When we look at the PD biomarkers, as I mentioned earlier, we're monitoring several biomarkers from the disease pathway. Here is the example of a 3-hydroxypropanic acid concentration at baseline. So due to the disease heterogeneity and rarity of disease, the evaluation of PD response is very challenging. The 3HP concentration at baseline before the treatment, you can see the huge inter-subject and intra-subject variability. So we also observed this from our natural history study. The biomarker for 3HP is chosen here as a PKPD modeling example because we also observed the metabolic decomposition event correlation with the 3HP concentration. This one's showing the 3-hydroxypropanic acid concentration change with treatment. On the right side, if you can see this, the figure x-axis is the time in days post-treatment, and the vertical line represents the dosing event. Y-axis is the percentage change of each subject post-treatment, because as you see early on, we're not taking overall 3P of all the subjects. We're rather looking at each subject, how their 3P change post-treatment only compared to themselves. So cohort 1 and cohort 3 have a significant 3P reduction post-treatment, while on the subject number 5 at second cohort has not responded to the treatment, but this subject might have a mild phenotype since there has not been any metabolic decomposition event observed 1 year prior to the treatment, also no MDE 1 year post-treatment. Our population PK/PD modeling is use 3HP reduction as our PD response. I want to also mention that our target, right? So for the natural history study, we observed PA patient after liver transplant, their 3HP reduction is about 70%. This is a goodness fit of PD response. To avoid over-parameterization, we're using the ratio to baseline as PD response, similar as the percentage change from baseline, but for each subject, they calculate their own response. So there are no need to estimate the baseline value since all subjects will start with 1 as baseline. We also included age as a covariate on the protein synthesis and protein degradation rate constant. PKPD modeling is continuously performed in the ongoing phase 1 clinical study to inform dose regimen for the subsequent cohort. The simulation results was utilized to identify lower peak to trough ratio of 3HP and a steady state 3HP concentration. So the current model predicts a greater and more stable 3HP reduction at a steady state with once every 2 weeks dosing frequency, with the emerging clinical data, all cohort 1 patients who were original on once every 3 weeks were offered to switch to every 2 weeks. And the metabolic decomposing event frequency reduction also confirmed that Q2W is better than Q3. So this is more of showing like we use PKPD model to guide the dosing frequency, and clinical endpoint also observed an improvement. To give a brief summary, population PKPD modeling and simulation have been applied successfully for dose regimen optimization, and the model structure depends on available data. Modified compartmental models could describe mRNA PK pretty well. In general, in indirect response model can be applied to the PK/PD relationship. As also early on, Tao mentioned that because the mRNA needed to synthesize protein, so there's a delay. So the model structure of the lipid nanoparticle encapsulated mRNA is generated similar to the lipid nanoparticle encapsulated siRNA. There are some challenges. When plasma PK is not translatable for PD, the tissue PK/PD data might be essential. As you all know, it's relatively easy to say to collect a tissue sample in preclinical setting, but in clinical study, it's normally not accessible. Model construction strategies presented are mostly empirical, fit for purpose, but not extensively mechanism-based due to knowledge gaps and data limitations. Physiological information on mechanistic components is lacking, so mechanistic modeling and applications limited. Also, the biology knowledge of disease pathway and target could be very helpful to build a better PK/PD model. With that, I'll turn back to the Tao, I think. Thank you. Now we can start the penalty, right? The panelists are all joined. I think I turn back to someone. Okay.
Yeah, so now let's start a panel discussion for Q&A. So we have 3 additional panelists here. Jesse, currently an executive director, translational medicine at Merck, and Manuela Brauer, senior DMPK project lead from the DMPK department at Bayer, and Yun Liu, a scientist at DMPK department from Moderna, in addition to all the speakers. Let me see.
You having an issue with audio or?
I guess let me see if we have any questions.
There's
one from Viva. Is small pack size not recognized by immune system as a red flag compared to the pegylated protein?
No questions from the audience, but you can ask them first.
I guess let me rephrase the question. Is small pack size from the LNP not recognized by immune system as a red flag compared to pegylated protein? Manuela, do you want to take this question?
I can take this question. I assume that the packs that are used for proteins are much larger, and these are usually the branch packs which have much more which look somehow like a tree, and probably the structure of the packs also plays a role in regard to recognition by the immune system. But perhaps in future we may also see more immune reactions also to the small size pack if you have more programs.
Okay, thank you, Manuela. I can also answer that question from clinical perspective. So for the first proof siRNA patisiran, which is also lipid nanoparticle encapsulated, the immunogenicity incidence rate is less than 4% across all the clinical studies, and there's no impact of anti-PAC antibody on the PK, PD, efficacy, or safety. So I think so far, in terms of anti-PAC antibody from LNP's perspective, it's not a major risk, I would say, and the clinical impact based on the current understanding is just not significant.
Yes, I agree.
If there's no question, I would have a question for Pedro for the ddPCR. Although I think you generate so many droplets that increase your sensitivity, does this assay also have a downside compared to traditional qPCR? Is it more sensitive to matrix interferences? Can you maybe comment on this?
Marcos. Well, I mean, in the ddPCR, as I said, we have a mixture of thousands of droplets. So essentially, you're basically diluting the potential effect of PCR inhibitors because you're less likely to have them on each droplet. But perhaps, I mean, the RT-qPCR still has the advantage on sensitivity, although with the ddPCR, you can also achieve very good sensitivities, very low copy number per total microgram per microgram of total RNA.
Thank you.
I think there's a question from the audience. So the question is liver the only or the main targeted tissue, or is there any other tissue that can be targeted with LNP? Yun, can you take this question?
Yes, I can. Yes, I can take that question. That's actually a great question. So for the most LNPs that currently used in preclinical stage or clinical stage, the liver is the target organ. Like most LNP deposits in liver, but there are also other tissues will uptake LNP as well. There are also some research in the discovery stage, like they are using some targeting moieties to modify the surface of LNP to enable the targeted delivery of LNP to other organs like lung or spleen after intravenous injections.
Thank you, Yun. There's another question about the double peak phenomenon. So the question is, for the second local peak of plasma mRNA in patients, is there any hypothesis on why it is that? I guess Yun, I'm sorry, Ming, can you take that question?
Yeah, so sorry, Yun and I in the same room. So the second peak, the local maximum which we observed, we believe is due to the redistribution of the lipid nanoparticle. Since we observed this in several of the study already so far, and this is not new because the siRNA, they also observe the same phenomenon. That's why I'm saying our compartment model is modified.
Yeah, I echo Ming's comment. Actually, you can check the population PK PD model for patisiran that has, I think it's also the same phenomenon, and it has been taken care of. Another question, can you measure or differentiate release mRNA versus encapsulated mRNA? Can you take this question, Yun? Yuan, can you take this question? How to differentiate released mRNA versus encapsulated mRNA? All right, I guess Yuan's probably having some internet issues.
Oh, sorry, we couldn't hear at beginning. So Yun is here. Can you say again the question?
Oh yeah, absolutely. So the question is, can you measure or differentiate released mRNA versus encapsulated mRNA?
Yeah, I think that's a analytical questions. I think we can definitely do that when we characterize the mRNA before the injections, like if there's any free mRNA that not encapsulated in the formulations. But once it's injected, say it's IV injected into preclinical models or patient, I think that's we need specific assays to differentiate like the mRNA that released from the particles and the encapsulated inside the particles. Basically, due to the instabilities of the mRNA, we think when the mRNA has been released from the particles, the half-life of the free mRNA would be very short in serum and tissues.
Thank you, Yuan. I think we are at the top of the hour, and thank you all for your attention, and we will see you next time. Bye.
Thank you very much.
Thank you. Bye-bye.
Thank you.